Hello, I'm Rachel Kolodny from the University of Haifa in Israel, and I want to thank the organizers for letting us present this work. So what we're interested here is in having a global view of the protein universe, a roadmap, if you will. And this is obviously something that we would really like to have, even like, for example, in today's keynote talk, Michael Levitt mentioned how interesting this would be. If we could have this roadmap, we could use it to better understand protein evolution, maybe search proteins better, maybe understand something about how we can do design or engineering. So this would be a very useful thing. The problem with this, the challenge, is that what we have are glimpses of local relationships among proteins. We don't have the global view. We have to construct it from these glimpses. Okay, so this is a very fundamental question, and this was looked at by many scholars before us. So I'll just mention quickly some work that was done before, just so you'll have like a view of who's who in this field. So we start from the PDB. We're interested in protein space of the proteins that we know their structures. And common wisdom says that the evolutionary unit are the domains. So we use domains rather than PDBs. So the very basic picture, roadmap that we have of the protein universe are the classification. And the classics are, of course, SCOP from the Merzen lab and CAT by Christina Orengo, and now there's ECOD from the Grecian lab. And these classifications, they organize all proteins into groups. For example, in CAT, they first say that there are 3 high-level groups, the all alpha proteins, the all beta proteins, and ones that have both alpha and beta. And then they further separate these into more groups and so forth. Within the view of classification, something very fundamental about it is it views protein space as island-like entities. Like there are these folds where they're similar to each other and not similar to the others. Of course, it has been recognized for a long time now that there are many cross-fold similarities, and these classifications have been modified. For example, now there's SCOP2 that takes account of these cross-fold similarities. But still, within this idea of classification, protein space looks like islands, entities that are different from one another. Alternatively, scholars have created maps of protein space. So in maps of protein space, you represent each protein domain as a point in 2 or 3-dimensional space, and they're placed such that the distance between 2 points somehow encodes the distance between the proteins. So this was first done by Orengo et al., and then Holm and Sander. Then there was a series of paper by Sang-Ho Kim and his students, where they added this level of coloring the points, in this case by the SCOP class. And then you can see that you can use your cognitive abilities to look at these maps and understand something about protein space. And even we had a paper a few years back where we looked at the functional diversity within these maps. The fundamental problem with these maps, when you embed the proteins in Euclidean space, is that you get the transitive property built in. That is, you can't have a situation where protein A is similar to protein B B is similar to protein B is similar to protein C, but A and C are very different. And this is a problem because Bastola et al. Showed that there are many, many cases where this is actually true, that transitivity doesn't carry over. So this is actually the problem with embedding these maps in Euclidean space. So what you're left with is using networks. So in networks, what you do is you represent every domain as a point, as a node, and you connect with edges to domains that are related to each other. And there's many works, I'm just mentioning a few. There's one by Golan Yona, who Michal was involved in. Then Dokleyan and the Shaknoviches had the PDUG, the protein domain universe graphs. Zhang et al. Had one. Skolnik created such a network where he connected 2 protein domains that were structurally similar. So the networks, in some sense, what they've shown, and in particular the network by Skolnik and co-workers, was the continuous view, the absolute other direction from the classification, where they showed that everything is connected to everything. So these were the 2 ends. And I didn't mention all the studies, but we surveyed them in a paper a year ago. Okay, so what we did is we looked at protein space. We took a large collection of protein domains. Here for the cartoon, I just drew 5 of them. And we compared all against all. Okay, because it's symmetric, it's sufficient to look only at the upper part of the matrix. And in every instance where we saw that 2 domains were similar, that is, they share a significant subpart. For example, you see the first domain and the third domain share this orange triangle. So I marked it with an asterisk. So in every such instance, we marked this in our matrix. And from this similarity matrix, we created a network graph. What is this network graph? Every domain is a node, and two nodes that share a significant part have an edge between them. What we actually did was we considered almost 10,000 sculpt domains representing all of protein space. We took a 70% non-redundant data set, and we created this graph. Okay, once we had this domain graph, we represented it just simply as nodes, and we colored the nodes based on their sculpt class. That is, if they're all alpha, we colored them in blue, all beta in red, alpha/beta in green, and alpha plus beta in yellow. In a way, what we're encoding is their biophysical properties. Okay, so here's an example from our actual graph, just to give you a sense of what places an edge between two domains. So you can see that D1VLUA and D1020A underscore share 241 residues, that's the orange parts, that can be superimposed to 1.6 angstrom RMSD, and they have a 57% sequence similarity. These are pretty similar subparts, okay? And indeed, this was the network that relates to this piece. Okay, so in the example I showed here, what we considered similar between two domains is if there were at least 75 aligned residues, the RMSD over these superimposed residues was less than some 2.5 angstrom, and the sequence similarity was higher than 30%. Of course, you can use different thresholds, which we actually did. We considered alignment length of 55 rather than 75, 2 angstrom or 3 angstrom RMSD rather than 2.5, and sequence similarity that ranges from 30% to 50%. There is this question that we said if we identify, and the question is, how do you identify? This is an issue. And in this case, because we wanted to identify as many relationships as possible, we used structural alignment program, and in particular, we used SSM by Krisnel and Henrik, because we wanted to use something that is very sensitive, to identify as many possible similarities among the proteins. But once we identified the similarities with the structural alignment, we enforced a sequence similarity threshold of 30% or 40% or 50% sequence similarity over the aligned residues. Now, of course, if you vary the thresholds, you would get very different networks. If you use very lax threshold, you would have a very connected network, like on the left. So for example, this network, the thresholds were 55 residues at most 2.5 angstrom RMSD and more than 30% sequence similarity. And if you use very strict thresholds, for example, 75 residues alignment length and more than 50% sequence similarity, the network will disintegrate. And if you use even more strict thresholds, eventually every node would be connected only with itself, right? This is a trivial thing. So indeed, what we saw that if we use not so strict thresholds, that is, if we use 50 residues, 3 angstrom RMSD, and 25% sequence similarity, then protein domain space is essentially a connected component. So this is a refinement of what Skolnick said, because he used essentially a 0% sequence similarity. When using only structural similarity enforcement, this is what he said. Okay, so one way to go about this, so the question is which thresholds should one use? And there's actually two ways to go about it. One is to search for the so-called right thresholds, and I'll get back to that in a minute. But I want to point out that another way of looking at it is thinking of using more strict or less strict thresholds as tuning the evolution dial. Like if you use very non-strict thresholds, very lax thresholds, it's as if you're looking further back in evolutionary time. And as you're using more and more strict thresholds, you're going closer in evolutionary time, and you're using only present-day information. So by looking at what happens when you vary these thresholds, this could give you hints to protein evolution. This was the art. But still, we wanted to have Still, we wanted to try and find somewhat right thresholds, and to do that, we calibrated them to fit Sculptfold classification. That is, we would like to create a network where the connected components are similar to Sculptfolds. So we considered a particular threshold combination. We looked at the network that was resulting from that particular threshold combination, and we measured how many times do we place things that are in the same Sculptfold in the same connected component, which percentage were successful in that sense, and which percent are we successful in not placing things that do not have the same Sculptfold, right? You don't want to err on either of these two. So I want to point out to you that placing a point in the upper left corner or in the lower right corner that is connecting everything, like completely being successful in connecting things that should be in the same fold is very easy. You just relax your thresholds and you get a completely connected component. And every time two folds are in the same Sculptfold, they will be in the same connected component. Similarly, if you want to be successful just in not placing things that do not have the same Sculptfold, that is very easy too. You just vary your thresholds to very strict ones, and you never, right, because it's essentially a disconnected component, it never happens that two proteins that are in different Sculptfolds are in the same connected component. So the trick is to get a combination of thresholds that will place it in the upper right corner. Right, this is the challenge. And as you can see, the diamond in pink does a pretty decent job at it. This is the 2.5 angstrom RMSD threshold, 75 residues, and 30% sequence similarity. So we used that one, and we created this network. And when we looked at this network, we were actually kind of excited. Like, I hope you guys are looking at it and are just as excited. So what we saw is that in this discussion, whether protein space is connected, continuous, or discrete, that is, has these island-like properties, it's actually both. So there are regions of protein space, for instance, the ones here, where it's really islands, exactly what the classifications tell us. And there are regions up there, which are mostly the alpha/beta proteins in green, which are very connected. Everything is kind of connected to everything, which is the people who are talking about the continuous nature of protein space, this is what they were talking about. And of course, all the examples were from there. So if I color this same picture based on the Sculptfold, and I just colored a few because otherwise it's too messy. So you see that there are definitely folds. For example, in blue, that's the globin-like fold, and it's a connected component. It's a very clear case. And, or the orange one here, or the yellow one here. But there are folds that fall within the connected component, the continuous region of protein space. These are alpha/beta proteins, and it's really connected. It's really a far more messier picture. So protein space is both. It's continuous and it's discrete. And it's actually, it's great to know that it depends where it's continuous and where it's discrete. It depends which type of protein you are. Okay, but we were concerned that maybe the issue was that we weren't selecting the thresholds properly. That is, if we kept varying the thresholds and the network would further disintegrate, it's just in a different pace, so to speak. That in the all alpha, all beta region, it was disintegrating properly first, and this is what we saw. And if we continued, we would disintegrate the alpha/beta into folds as we would expect. So we did this same test for the different Sculptfolds. And what I want to point out to you is the different Sculptfolds behave differently. That is, if you look at the all beta in the upper right corner, you see that the pink diamond does a very good job in separating it into folds. But in contrast, in the alpha/beta in the lower left corner, there is no combination that disintegrates it into fold-like islands, into fold islands. And another way of saying it is that when you vary the thresholds, when it disintegrates, it happens all over the place. It's not like you can first disintegrate the ones connecting between folds and then only within folds. It disintegrates all the way through. And the alpha/beta are like that, and to some extent the all alpha. And this is different from the all beta and the alpha plus beta domain that tend to be ordered within these classification-like islands. Okay, so the 1 minute, seriously? Okay, we have a problem here. Okay, so I'll just say this. We looked at these networks a lot. To do this, we used the networks presented within the Cytostruct network visualization tool, which we think is super cool. We thought it was so cool that Sergei Nepomniachtchi created for it a plugin, which is called Cytostruct, which we just published in Structure, and I think each and every one of you should go to the website, download Cytostruct, install it, and then you can see all our data. It's all available online. It's really quite cool, because what it does is it overlays on top of the visualization tool of the network the ability to see the structures within a molecular viewer. You're probably thinking, which molecular viewer? That's just it, any molecular viewer. PyMol, VMD, UCSF Chimera, JMol, anything that you want. And you can do it with this tool, and it's not just, you see here, this is an example, and you can use all the abilities within the molecular viewer. In our case, it was superimposing the structures using CABJ. It's all done for you through the tool. It's really cool. You can look at these things, and you see, you can connect it to any node or any edge within the network. It's really, really cool. You can configure it, you can download it, because it's really a click of a button. And then once you do, you can see our demos. We have a website set up for it. And you can, not only can you see the demos, you can download the code. It's all open source. I'll just give a shout out of 2 examples, because we used it for visualizing these universe of proteins domain networks. But you can use it for anything, because it's such a general purpose tool. For example, we used it in Contemplate. Aya Narunsky used it, where she's generating, given a query structure, alternative conformations, suggested alternative conformations from the PDB, and it's all within your web browser. Or we did a protein-protein interaction server where we converted PISA output into these networks that you can see the visual thing. Okay, so I just want to say one last thing, really, one last thing. So what we showed here, that if you have the similarity information within this matrix, you can create the similarity network, which is what we did. But actually, what would be very interesting to do is to take this matrix and extract from it the recurring elements, the motifs, so to speak. And for these motifs, right, for example, in this case, we see that there's the triangle and the diamond and the purple heart and so forth. We would like to create a network where you have for each motif a node, and when 2 motifs co-occur within a domain, you connect them by an edge. This is the dual network of the similarity network that we looked at before. But it would be nice if we could identify these motifs from this similarity matrix. In particular, in this instance, we would probably not be able to identify the square and the heart, because they do not repeatedly occur. They occur only once. Okay, so we looked at it somewhat in the paper. We saw that recurring motifs are very common in protein space, but it would be very interesting to characterize these. Okay, so let me summarize and say that I think that domain and motif networks are a wonderful way to study the protein universe. We saw that it's both continuous and discrete, which is cool. And the continuous regions are mostly the regions where the alpha/beta domains are. Cytoscape can help here and elsewhere. And we hope to use it further. And I'll just say that I did this, it's not me, it's Sergei Nepomniachtchi, who is in NYU Polytechnic Institute, and Neil in Tau. It was.
We have time for a couple of questions, most. Please. And then,
yeah, so it's not so much as a question as a Anregung. I lose my English. Basically, structure space, in at least large parts, seems to be continuous. I agree. Surprisingly, sequence space in some parts also has elements of continuity. However, my experience would suggest that sequence structure space, the correlation of sequence space to structure space, is substantially discontinuous. And so in the map that you showed, I think it would be tremendously interesting to map sequence similarity, for example, by color, onto the structural similarity you have, because I think large parts of this will turn out to be polyphyletic.
That's an excellent idea.
Right.
That's an excellent idea.
next question.
Have you thought about treating edges as weighted? And that way, you'd have to take all your various metrics and somehow come up with a single number. that way you won't have to use threshold. The visualization would take account of the edge weights to, push nodes further or closer apart. And
That's, in a sense, this is what Dr. Lupac said, is that if I could encode into the edges the sequence identity or similarity between them, so the overall measure is somehow captured when we vary them. So we see that maybe it would be cooler if it would be just by weight, but if we vary it, the overall measure is somehow captured when using different thresholds. But if we could overlay information about only one aspect, like, yeah, we'll try that.
The very last quick question. Yes.
Very nice talk. My question is about if you could comment on why do certain folds, when you tune the thresholds, why do certain folds perform better than others?
It's not better, it's just different.
Okay.
It's a good question. Like, I think if we could, so Charlotte Dean has work that shows that in somewhat similar networks, the ones that are more in the middle are older ones. So she correlates the age with the being in the middle, which this would be fabulous if this, like, if we could show this from, like, so it could be time, it could be their properties, like maybe the alpha/beta have, like, they're more fit, like they're just better at this, at composing with many different, like, I don't know, you have an opinion? It's a good, I don't know, it's like the, in some sense, the holy grail of understanding what are the forces at play. Like, is it just time or is it the biophysical properties? Like, it's, like, this is what we, so apparently some think it's a biophysical properties, but this is like, this is the reason we bother with this whole business. We care about this.
Okay.
Thank you.
So let's close this session by